Assalamu alaikum everyone. So today we are going to start with our chapter number 2, reproduction in lower and higher animals, part 11. So today we are going to discuss regarding the embryonic development. So starting with the zygote, as you all know what do you mean by zygote. Zygote formed as a result of syngamy. It is activated to divide. Okay. It is formed because of the result of syngamy and it is activated to divide, and after division it will form in the form of that is called as what? Cleavage. The cleavage will form. Here we will discuss the process of cleavage and the formation of blastula. Okay. So under cleavage, it is the process of early mitotic division of the zygote into a hollow multicellular blastula. What do you mean by cleavage? It is a mitotic division, and it is the early mitotic division of the zygote in which zygote will multiply in a hollow multicellular blastula. It does not involve the growth of the daughter cells. The cells formed by cleavage are called blastomeres. It does not involve the growth of the daughter cells. In this, the growth of the daughter cell will not take place. Instead of that, the cells formed by cleavage, they are called as what? Blastomeres. Since there is no growth phase between the cleavages, the size of blastomeres will be reduced with every successive cleavages. Okay? The size of the blastomeres will be reduced. It will be go on reducing with every successive cleavage. With every successive cleavage means when there is a 2-cell stage or 4-cell stage or 8-cell stage, it will go on reducing with the size. As the successive cleavage goes on moving on, so the size of the blastomere will be get reduced. As the size reduces, the metabolic rate increases. Subsequent cleavages are thus faster than the earlier one. This requires rapid replication of the DNA and high consumption of oxygen. All of these, it requires what? Rapid replication of DNA. Due to this, the replication of the DNA will become very fast, and high consumption of oxygen. They require a very good amount of oxygen. They will consume the oxygen in a very good quantity. All this happens in the process called as what? Cleavage. The process of cleavage takes place in this manner. Now, coming to the process of cleavage, in human, the cleavage is holoblastic. Now, what do you mean by holoblastic? The whole zygote gets divided. The whole zygote, it will get divided. The cleavage planes may be longitudinal or meridional, and equatorial or horizontal. All these are the different positions of the cleavages, which are called longitudinal or meridional. Meridional is also the term which is also similar, which is also used for longitudinal, and they are also equatorial or horizontal. Equatorial and horizontal are one and the same word, and longitudinal and meridional, they are the one and the same word. So, it is radial and intermediate. It is radial and intermediate. That is, fate of each blastomere is not predetermined. Fate of each blastomere, it is not predetermined. It is not basically pre-planned. Okay. The first cleavage in the zygote is meridional, also known as longitudinal, and they occurs at about 30 hours after fertilization. They occurs about how much hours? 30 hours after fertilization. It divides longitudinally into two blastomeres, one slightly larger than the other. The second cleavage is also longitudinal, but at the right angle to the first one, and occurs after 30 hours of first cleavage. The third cleavage is horizontal. After the third cleavage, the embryo is in 8-cell stage. All this you all can see in the given picture, in the given figure over here, and you can understand how the cleavage formation takes place and how the zygote develops into the 2-cell stage and into the 2-cell stage or a 4-cell stage or the 8-cell stage. Okay. Now, as the cleavages are going on, the young embryo is gradually being pushed. The young embryo is gradually being pushed towards the uterus. Then, by the end of the 4th day of fertilization, after fertilization, embryo is a solid ball of 16 to 32 cells and externally looking like mulberry. How it looks? It looks like a mulberry, and it will be a solid ball-like structure, about 16 to 32 number of cells by the end of the 4th day after fertilization, and externally looking like a mulberry. This stage is thus called as what? Morula. It is called as what? Morula. So basically, what do you mean by morula? It is a mulberry-like structure, and basically it is an embryo with a solid ball-like of structure, which contains a number of cells of about 16 to 32. Okay, this is called as what? Morula. The morula shows cells of two types. It contains two types of cells: smaller, clearer cells towards the outer side, and inner cell mass of the larger cells. The cells are compactly arranged. They are over here in morula. The cells are compactly arranged. Till the formation of the morula, the zona pellucida is retained around the embryo, and thus there is no change in the overall size from zygote to morula. Just because of the zona pellucida, it is retained around the embryo, and thus there is no change in the overall size from zygote to morula. The morula reaches the isthmus and gains entry into the uterus by the end of the day 4. So this is what all happens after the 4th day after the fertilization. How it goes and how it reaches to the isthmus, and then how it gains the entry into the uterus by the end of the day 4. Now here we will study in the next stage, that is called as what? Blastulation. Blastulation is the process of formation of the hollow and multicellular blastocysts. It is the process of formation of the hollow and multicellular blastocyst. The embryo, which is now called as blastocyst, that enters the uterus, here it enters in the uterus, remains floating in the uterine cavity for about 2 to 4 days. After its entry, that is till the end of 7th day after fertilization, the outer layer of cells seen in the morula now form the layer called as what? Trophoblast. Okay. Then cells from the trophoblast, they begin to absorb the glycogen-rich uterine milk. Cells of the trophoblast, they begin to absorb the glycogen. Glycogen is one of the form of the saccharides. It's one of the form of the carbohydrates which are rich in the here. Thus they begin to absorb the milk, that is called as what? Uterine milk, and they are glycogen-rich milk. They are rich in the glycogen. Okay, uterine milk. So cells from the trophoblast, they begin to absorb the glycogen-rich uterine milk. The blastocyst doubles in size due to the absorption of the glycogen. The blastocysts, they get converted into double in size from 0.15 mm to 0.30 mm. Okay, with more fluid entering inside the blastocyst cavity. Inside the blastocyst, cavity is formed. Just because of the more fluid entering into the blastocyst, cavity is formed. These outer cells become flat. These outer cells, they become flat and are called trophoblast cells, since they help only in absorbing the nutrition for the developing embryo. So they are called as what? Trophoblast cells. The inner larger cells from inner cell mass or embryoblast, the embryo proper develops from the embryoblast. These remain attached. Okay. What happens over here? It remains attached to the trophoblast on only one side. The trophoblast cells in contact with the embryonal knob, and these knob are called as cells of Robber. Cells of Robber. At this stage, the blastocyst shows polarity. The side with the inner cell mass is called the embryonal end, and the side opposite to it is the abembryonal end. By the end of the 7th day, the blastocyst is fully formed. By the end of the 7th day, the blastocyst is fully formed. It is now ready for implantation and gastrulation. Now it is ready. The blastocyst, by the end of the 7th day, it will be ready for implantation and for the further process that is called gastrulation. The function of zona pellucida is to prevent the implantation of the embryo at an abnormal site. What is the function? Many times it happens that it will come for the 1 mark question also, that what is the function of zona pellucida. So the function of zona pellucida is to prevent the implantation of the embryo at an abnormal site. Otherwise, it will be very harmful for the upcoming embryo or the zygote. It does not expose the sticky and phagocytic trophoblast cells till it reaches the implantation site, that is within the uterus. Hence, zona pellucida now ruptures. So here, what it happens over here. Here you can see how it happens and what happens over here. Now coming to our next topic, that is called as what? Implantation. The blastocyst after its formation gets implanted or embedded. Implantation means getting embedded or getting perfectly attached to something. That is called as what? Implantation. So the blastocyst after its formation gets implanted or embedded into the endometrium of the uterus. Into the endometrium wall of the uterus. The blastocyst will be get formed and it will be get implanted into the endometrium of the uterus. This process usually begins on the day 7 after fertilization, and by the end of the 10th day, the embryo is completely buried inside the endometrium. By the end of the 10th day, the embryo is completely buried inside the endometrium. The embryo usually implants in the region of the fundus of the uterus. This is the actual position where the embryo gets implanted. So it will be get implanted or embedded in the region of the fundus of the uterus. All these are the different parts of the uterus, that is called as what? Fundus. In the process, the embryo attaches itself by its embryonic pole close to the endometrium. The trophoblast cells of the animal pole have the power to stick to the uterine wall. Rapid division of the trophoblast cells lying against the embryonal knob takes place. It results in the formation of two distinct layers, and these layers are called as what? Syncytiotrophoblast or cytotrophoblast. So all this happens into the implantation process. Coming to our next topic, that is called as what? Gastrulation. It is the process of formation of gastrula from the blastocyst. In the gastrular stage, here the rate of the division or the rate of the cleavage will be slowed down. It will slows down the rate of cleavage or the division. Okay, and it will occur after the 8th day of the fertilization. The gastrulation will take place after the 8th day after the fertilization, on the 8th day after the fertilization. Okay, then there are two important events that take place actively for gastrulation process. First one is differentiation of blastomeres, and second one is morphogenetic movements. These are the two important events that take place during the time of the gastrulation process. Starting with the first one, differentiation of blastomeres. How the blastomeres get differentiated. This process results in the formation of three germinal layers, that is ectoderm, mesoderm and endoderm, from the cells of the embryoblast. So here how the differentiation of the blastomere takes place by the formation of the three germinal layers, that is ectoderm, mesoderm and endoderm. Coming to the next one, morphogenetic movements. These are the different types of movements to reach their definite place in the embryo. What? These are the proper morphogenetic movements, or these are the proper different types of movements to reach at their definite place in the embryo. Coming to our next slide, cell on the free end of the inner cell mass, they are called as what? Hypoblast, which is also called as, which is nothing but it is a primitive endoderm. The previous or the older one, embryo, endoderm. They become flattened. They start dividing and grows downward towards the blastocele. Blastocele means a cavity of the on the blastocyst, that is called as what? The cele. Cele means cavity, where it is in the blastocyst, therefore it is called as what? Blastocele. Cavity of the blastocyst. This layer called endoderm is first to differentiate. It grows within the blastocele, and forms a sac-like called structure, and that is called as what? Yolk sac. The remaining cell of the inner cell mass in contact with the cells of the robber are called epiblast, which is a primary ectoderm. Both layers form a flat bilaminar embryonal disc. Cells of the epiblast divide and redivide and move in such a way that they enclose the amniotic cavity. That they enclose the amniotic cavity. The floor of this cavity has the embryonal disc. Next is the pyramidal cells of the disc towards the amniotic cavity from the embryonal ectoderm. It will form the embryonal ectoderm. The roof of amniotic cavity is lined by amniogenic cells. Later, these cells divide and redivide to form the amnion. It will divide and redivide to form the amnion. Amnion is an extra embryonic membrane that surrounds and protects the embryo. What do you mean by amnion? It is the extra embryonic membrane that surrounds and protects the embryo. Basically, its function is protection of the embryo. As a result of all these changes, the bilaminar embryonic disc is positioned in between amniotic cavity and yolk sac. Now, actual gastrulation occurs about 15 days after fertilization, in which the bilaminar embryonic disc is transformed into the trilaminar embryonic disc. This transformation occurs by division, rearrangement, and migration of cells it occurs by division, rearrangement, and migration of cells of epiblast. It begins with the formation of primitive streak and a shallow groove on the surface, which is called as primitive groove. Next is, this streak progresses from posterior to anterior end of embryo. From site of a primitive streak, a third layer of cells called mesoderm extends between the ectoderm and endoderm. Anterior end of primitive groove communicates with yolk sac by an aperture called blastopore, which will be a future anus. The embryonal disc now has differentiated into three layers: ectoderm, mesoderm, and endoderm. The further process after gastrulation is called as what? Organogenesis. Now, coming to the last slide of our today's lecture, the fate of germinal layers. There are three different layers of germinal layers. There are three different germinal layers, and they are called as what? Ectoderm, mesoderm, endoderm. We will differentiate all these three. Coming to the first one, ectoderm. Ectoderm give rise to epidermis of skin, hairs, nails, sweat glands, salivary glands, mammary glands, lacrimal glands, sebaceous glands, cornea, lens, retina, conjunctiva, nasal epithelium, enamel of teeth, internal and external ear, foregut, hindgut, adrenal medulla, anterior and posterior pituitary, pineal gland, entire nervous system. All these are the different parts which come into origin, which brings about just because of the ectoderm. Ectoderm give rise to all these different parts of our body. Epidermis of the skin, upper layer of the skin. That is called as what? It means what? The upper layer of the skin. Hairs. Epidermis of the hair means upper part of the hair, upper part of the nails. Sweat glands. The glands which secretes the perspiration, which secretes the sweat. Salivary glands, which secretes the saliva. Mammary glands, which brings about the milk at the time of the parturition, just after the parturition, after the delivery. Okay, then sebaceous gland. It gives out the sebaceous. Okay, they are present in our skin, sebaceous glands. Cornea, it is part of the eyes. Lens, it is also is parts of the eyes. Retina, conjunctiva. All these are the different cornea, lens, retina, conjunctiva. All these are the different parts or the portion of the eyes. Nasal epithelium means the nose epithelium, the nose, outer covering of the nose. Enamel of teeth, the outer covering of the teeth. Internal and external ear, the internal part of the ear and also the external part of the ear. Then foregut, hindgut. All these are different portion of the intestine. Adrenal medulla, this is the part of the brain. I'm so sorry. Adrenal medulla, it is a part of the kidney medulla, the middle portion of the kidney, renals. Okay, above the kidney there is adrenal gland, and that adrenal gland, it contains the medullary part, the middle portion part. So that also comes into origin just because of the ectoderm, or ectoderm makes the adrenal medulla also. Then anterior and posterior pituitary gland. Pituitary gland is the one of the master gland. Earlier it was called as was a pituitary gland is the master gland, which is situated in the midbrain. Then pineal gland, entire nervous system. All these are the different portion. All these are the different parts of our body, which is just originated, which comes into existence just because of our ectoderm. In short, I just want to say that ectoderm give rise to all these different parts of our body, what I have just said right now. Coming to the mesoderm. Mesoderm forms all types of muscles except the iris muscles and ciliary muscles of the eye, which originate from the ectoderm. Iris muscle and ciliary muscles, these are the muscles of the eye which they originate from the ectoderm. Otherwise, all the different types of muscles, they originate from the mesoderm. Basically, this is the middle layer. Then connective tissue is also formed from the mesoderm. Dermis of the skin, adrenal cortex. Now here adrenal cortex, the renal cortex. Then heart, blood vessels, lymphatic vessels, middle ear, dentine of the teeth, the proper arrangement of the teeth, urinary and reproductive ducts, gonads, kidneys, sclera and choroid of the eye. All these are the different parts or the forms of the all these are different parts of our body which forms due to the mesoderm, or mesoderm forms all these different parts of the body. Coming to the last part of our today's lecture, that is endoderm. Endoderm develops into epithelium of midgut, glands of stomach and intestine, tongue, tonsils, lungs, trachea, bronchi, larynx, urinary bladder, vagina, liver, pancreas, thyroid gland, parathyroid gland, thymus gland, Eustachian tube. Eustachian tube, basically they are situated in the ear. And epithelium of the urethra, lining of the middle ear. So these all are the different parts of our body which comes into existence, which develops due to the endoderm. Thank you all. So now in the next lecture I'm going to discuss all the remaining part of our chapter. Thank you so much.